Mosquitoes, biting flies, and internal parasites are a pain. Did you ever wonder about their real effects on human society? In this lecture, we'll explore adaptations of some of the most economically important animals on our planet, invertebrates that have adverse effects on humans. Locusts and other invertebrate creatures devour human crops, and mosquitoes and other biting invertebrates deliver parasites and disease into humans. Both of these adverse impacts cost human society billions of dollars and millions of human deaths per year. Before we go any further, I should warn you, you don't want to eat while watching this lecture. Put your snacks down. Are you ready? Okay, let's first ask a fundamental question. Who are the baddest, ugliest invertebrates? We find our world's creepy crawlies so creepy because they're small, different from us, and they bite us, parasitize us, and terrorize us when they swarm. Locusts, flies, mosquitoes, fleas, cockroaches, ticks, spiders, worms, and tiny internal parasites all meet the common notion of bad and ugly. But do you know what the deadliest animal on Earth is? I'll wait. Believe it or not, it's the lowly tiny mosquito. According to the World Health Organization, mosquitoes spread malaria, West Nile virus, yellow fever, dengue fever, and several other diseases, killing more than 2.5 million people per year. Why do mosquitoes bite people, you might ask? Mosquitoes and other biting flies sense the world differently than we do. Mosquitoes can detect the carbon dioxide we give off when we are breathing, as well as our mammalian body heat, and they are very attracted to both. The female mosquitoes are the ones who bite us, but both sexes have exquisitely sensitive sensory systems. In fact, a male mosquito, using its antennae, can hear the whirring of a female mosquito's beating wings 1/4 of a mile away. Each kind of mosquito is active at species-specific times of day, usually dawn and dusk. Mosquitoes are also affected by ambient temperatures, amount of light in the environment, and even the amount of moisture in the air. Many mosquitoes are only found at certain heights above the ground, the better to find victims. When you are outdoors when they are active, you are likely to be bitten unless you wear protective clothing and insect repellent. Malaria, carried by the Anopheles mosquito, is the main reason that mosquitoes are considered the deadliest of all animals. More than 400,000 people die of malaria each year, although hundreds of millions more suffer from malaria annually. More than 40% of the world's population lives in areas occupied by Anopheles mosquitoes, and most of those affected are African children under 5 years of age. 1 child dies of malaria every minute, according to officials at the World Health Organization. But its victims aren't limited to Africa. One of my uncles was afflicted for years after World War II, following his service in the South Pacific. Even today, good friends of mine have been affected by malaria. It is an awful disease, causing fever, chills, and weakness that seem to clear up but always come back, like getting the flu over and over and over again. In some cases, it causes liver failure and death. Malaria organisms are unicellular eukaryotes that are internal protozoal parasites in humans. From the phylum Apicomplexa, class Echinodaceta, related to the organisms that cause babesiosis in cattle, coccidiosis in birds, and toxoplasmosis in any mammal, including humans. The malaria parasite is probably the best known of these apicomplexan protozoans, protozoans being single-celled parasites. The protozoan parasite that causes malaria is a Plasmodium species, which spends part of its life cycle in the mosquito after it is ingested by a mosquito, then migrates to the mosquito's salivary glands from where it is injected into humans via biting. After it enters the human ecosystem, The Malarian sporozoite, which is the motile infectious form that migrates to liver cells, penetrates them, and goes into an asexual reproduction cycle called schizogony, or multiple asexual fission, producing tens of thousands of myrizoids, which are the passive forms carried in the bloodstream. In the most common malarial organism, Plasmodium falciparum, these myrizoids enter the human's red blood cells after 1 or 2 weeks and undergo schizogony again. The malarial myrizoids in the red blood cells become amoeba-like trophozoites, the blobby eating forms that feed on hemoglobin. Sickle cell anemia, lower levels of normal hemoglobin, is a human adaptive response to living in malarial areas that confers resistance to malaria, although scientists are not sure precisely how this works. Sickle cell is most common in Black people of African descent, affecting up to 10% of African Americans. It affects about 20% of Africans living on the former so-called slave coast of West Africa, where many African Americans' ancestors came from. As each trophozoite undergoes asexual reproduction within the red blood cell, it produces a few dozen myrizoids, which burst out and infect new red blood cells. When a red blood cell containing myrizoids bursts, it releases the malarial parasite's metabolic byproducts, which have accumulated in the cell. Release of these damaged bits of foreign cells in the human blood circulation trigger a significant immune response that causes the chills and fever characteristic of malaria as we know it. The populations of malarial organisms maturing in the bloodstream are synchronized to some extent, so the episodes of chills and fever also have a periodicity that is characteristic of the species of Plasmodium, perhaps every 48 to 72 hours. After some cycles of schizogony in red blood cells, infection of new cells by some myrizoids causes production of gametocytes, which are malaria's equivalent to sperm and egg cells, instead of another group of myrizoids. This is the phase when the human host becomes infectious to mosquitoes. Then, following ingestion of these macrogametocytes and microgametocytes by mosquitoes feeding on the infected human, they change into mature gametocytes and fertilization happens. The fertilized protozoal cell in the mosquito gut is called an ookinete, which penetrates the mosquito's stomach wall and becomes an oocyst beneath the stomach lining. This oocyst produces thousands of sporozoites. Then the oocyst ruptures and the sporozoites migrate to the mosquito's salivary glands. From there, they are transferred to another human by the bite of that mosquito. Although there are treatments for malaria, the parasite evolves quickly, and over 70% of cases seem to be resistant to quinine and other treatments now. The debilitation of sub-Saharan Africa human populations from malaria reduces development and national productivity, so is a serious health problem. Increasing education about mosquitoes and the use of bed nets to prevent entry of mosquitoes so they cannot bite sleeping children, and increasing use of effective modern anti-malarial drugs helps to mitigate the widespread effects of malaria. So, we know that the tiniest creatures carry diseases to animals and people. Disease is spread not only by mosquitoes, but by tsetse flies that spread sleeping sickness and kissing bugs that spread Chagas disease. There are over 125 species of kissing bugs. Some are found in over half of the states in the US, and every single one can be host to the trypanosome parasites that transmit Chagas disease. Disease is also transmitted to us and other mammals by ticks and fleas. Lyme disease, caused by the spirochete bacterium Borrelia, is carried by ticks. Although this relationship of the disease to tick transmission wasn't proven by scientists until 1981. Even our household dogs are very likely to get Lyme in areas where Lyme is prevalent in the tick population. So they should be protected by effective drugs that can be prescribed by your family veterinarian. Fleas live everywhere there are other mammals. These small pests transmit typhus caused by rickettsial organisms, as well as bubonic plague caused by the bacterium Yersinia pestis. This is the Black Death organism that killed millions of people in the 14th century. But nothing really compares to mosquitoes. West Nile virus, Zika virus, and chikungunya are all examples of viruses transmitted to humans by mosquitoes. Since mosquitoes are arthropods, these are called arthropod-borne viruses, or arboviruses. Several viruses that affect humans are carried by day-active Aedes aegypti and Culex mosquitoes, which makes these mosquitoes doubly dangerous to day-active humans. Arboviruses can be transmitted by other arthropods as well. So you see why I include mosquitoes and some of their biting invertebrate relatives in my definition of bad invertebrates. As we discussed, there are good insects that we love, and now we have one more reason to love them. Some insects are enemies of mosquitoes that help in their control, for instance. Dragonflies and their damselfly relatives love to munch on mosquitoes. Although adult dragonflies only live a few months, their larvae can live underwater for years before they become adults. So adult dragonflies are eating up flying adult mosquitoes, while dragonfly larvae devour as many mosquito wigglers as they can find in the water. Yay, good bugs! Sometime around 200,000 years ago, our species emerged in Eastern Africa and eventually spread around the world. Until about 15,000 years ago, at the end of Earth's most recent ice age, humans had migrated to virtually every area on Earth where humans could survive, bringing some parasites with them and collecting others on the way. Microbiologists tell us that during our relatively short history on Earth, humans have acquired a huge number of parasites, about 300 species of helminth worms, and over 70 species of protozoa out of the tens of thousands of worm-like species on our small planet. Parasites that infect humans have been classified as either heirlooms or souvenirs. Heirlooms are considered those parasites that we inherited from our primate and human ancestors in Africa. And souvenirs are considered those that we have acquired from animals with which we have come in contact during our evolution, our human migrations, and our agricultural practices. Our human development of settlements and cities facilitated the transmission of infections from human to human. And when we opened up trade routes, it resulted in the wider dissemination of parasitic infections. The relatively recent Columbian Exchange, which flourished for 3.5 centuries from the earliest 16th century to the mid-19th century, exchanged parasites between the New World and the Old World. In recent times, the spread of human immunodeficiency virus, HIV and AIDS, and the immunosuppression associated with these conditions has apparently resulted in the establishment of a number of new opportunistic parasitic infections throughout the world. So this process of collecting parasites is still going on. We've learned a lot about the past history of parasitic infections from studies of archaeological artifacts, such as the presence of helminth eggs or protozoan cysts in coprolites. Those are fossilized or desiccated feces, and naturally or artificially preserved bodies. A new science, paleoparasitology, has emerged from these studies, and it has helped us to understand our long-time relationships with human parasites. Helminth is a general term for worm-like animals that are internal human parasites. They share body plans in that they are bilaterally symmetrical. Their sense organs and nerve centers are concentrated in the head, and they have a dorsal and a ventral surface. The flatworms are platyhelminthes, and the roundworms, or nematodes, that infect humans have adaptive anatomic features that reflect common physiological requirements and functions. The outer covering of all internal worm-like parasites is the cuticle or tegument. Flatworms have ways of holding on to their hosts. Ribbon-shaped tapeworms, the cestoda, have a head called a scolex that have hooks to embed in host tissues. Leaf-shaped flukes, the trematoda, have suckers for adhesions instead. Male trematodes of several species possess accessory sex organs that are external modifications of the cuticle. The internal alimentary, excretory, and reproductive systems of these creatures can usually be identified by an experienced observer. Tapeworms are gutless wonders. There are more than 1,000 species of tapeworms known to parasitologists. Like other cestode species, tapeworms require at least 2 hosts and are digestive tract parasites. Although Taenia saginata, the Greek word taenia means ribbon, is called the beef tapeworm, it lives as an adult in the human intestine. The juvenile forms live in the intermuscular tissue of cattle, and eating undercooked or rare beef is a way for humans to get this delightful parasite, which can reach a length of 30 feet or more in the human gut. The scolex of this organism cannot be considered a head. It is simply the anterior anchoring section of this modern parasite, and is apparently a leftover remnant of the posterior part of some ancestral form. In the beef tapeworm, the scolex has 4 suckers instead of hooks, and behind it is a short neck. The scolex is followed by a linear series of reproductive units called proglottids, and tapeworms have muscles, an excretory system, and sensory endings in modified cilia that inform a primitive nervous system. There is typically a germinative zone just behind the scolex where new proglottids form. The main body of the tapeworm is simply a chain of proglottids and is called the strobila. As new proglottids form in front of the individual proglottids near the scolex, the older ones move down the tape and their gonads mature. The beef tapeworm may have as many as 2,000 of these proglottid segments. Some zoologists suggest that this segmentation is best considered an adaptation to increase reproductive capacity via replication of sex organs, rather than true segmentation or metamerism that we observe in annelid earthworms. Mature proglottids carry infective, shelled larvae, which become detached and are passed in human feces to reinfect cattle and start the infective cycle all over again. When a person is infected with one of these tapeworms, numerous gravid proglottids are ejected daily, and sometimes they even crawl out the anus. Yuck. The entire surface of cestodes is covered with microscopic projections called microtrichas, which are similar to the microvilli in the vertebrate small intestine. These microtrichas are an adaptive anatomical advance for tapeworms, since they greatly increase the surface area of the tegument, and all nutrients need to be absorbed across the tegument as the tapeworm sits awash in the juices of our intestines. Eventually, the tapeworm's gravid proglottids break off and pass with feces. They then crawl out of the fecal mass and onto nearby vegetation. Remember, they do have muscle. There, they might be eaten by grazing cows. If they start to dry out, though, the proglottids will rupture and embryos will be distributed onto nearby grass and soil. These embryos can remain viable as long as 5 months outside a vertebrate body. Shelled larvae, called oncospheres, that are swallowed by cattle hatch and use their hooks to burrow through the intestinal wall to gain access to the blood or lymph system. From there, these microscopic creatures finally reach muscle organs where they insist to become cysticerci. These juveniles develop an invaginated scolex, but remain quiescent. When a human eats Raw or undercooked beef, the cyst wall dissolves, the scolex evaginates and attaches to the person's intestinal mucosa, and new proglottids begin to develop behind the scolex. In 2 or 3 weeks, a mature tapeworm forms and the cycle starts all over again. About 1% of American cattle are infected, and Americans eat a lot of beef. Although my family loves to eat rare beef, we can avoid infection by beef and pork tapeworms by thoroughly cooking our meat, or maybe by being vegetarians. I could talk about parasitic worm phyla for hours, but I'm not going to. Instead, I'd like to introduce you to Tim Walsh, a veterinary medical officer at the Smithsonian's National Zoo. I've invited Tim here to talk about one of the least known but most interesting departments at the zoo, the veterinary pathology department. Tim studies all sorts of parasites and the diseases they cause in our animals. Tim, I want to thank you for joining us today, and I'm going to start you off with a very, very important question. How often do you wash your hands?
Pretty much after every doorknob. Yeah, definitely. I mean, in my line of work, I definitely wash my hands a lot. I think it is one of those things where you have to be cognizant of what the realities of being a biological organism in the world is. There's always a potential for you to pick up something. If you're dealing with animals, especially, say, wildlife workers dealing with raccoons, they better wash their hands a lot. They better clean the environment a lot. There's lots of considerations.
Let's talk about that for a minute. You obviously have some special risks, but what are the most common parasites that our children, and in fact all of us, can get exposed to out here in the world? What are some of the big ones?
I think some of the most important things to keep in mind when you're dealing with parasites is that, one, each kind of parasite's a little bit different, and the hosts that they affect are a little bit different. And so probably some of the most important things to know when you're trying to deal with parasites in animal species especially is their lifestyles. For parasites, some of the basic lifestyles that exist is they can either be direct, meaning that they don't need more than one host to complete their life cycle, or they can have a complex multi-host lifestyle, and that's usually considered one animal is a definitive host in which they reproduce in, and another animal is an intermediate host in which they have to go through certain cycles to become infective for their definitive host. If we start going through the different classes of parasites, some of the main ones I think about are arthropods, which are the more insect-like ones. People know of ticks and mites and stuff. There's also protozoa, which are the single-cell organisms, very simple cell organisms, which are common infections, say, in the GI tract, like amoeba, or possibly less-known infections like toxoplasma, coccidia, sarcocystis, or malaria. Then we have the very common nematodes, which are roundworms. A lot of people know those from having pets with the roundworms that puppies are always treated for when they first get them. Then cestodes, which are tapeworms, trematodes, which are flukes or flatworms. So those are the main categories that we deal with. There are lots of different types, and the more zoologists look into the taxonomy and genetics of these, some of these are being separated out into different species like the myxozoa, so that starts getting much more complicated.
One of the better known ones, I think, for our audience is toxoplasmosis. Can you tell us a bit about that?
Yeah, toxoplasmosis is a protozoan from a group called the apicomplexans, and it's one of those that has a multi-host lifestyle. Probably one of the more significant or impactful parasites we deal with, in that in addition to the typical domestic cat being the main host, everybody has it, cats are the only ones that the parasite reproduces sexually in the intestinal tract, and so it's passed out in the feces. When those feces that contain the oocysts from the toxoplasma parasite are ingested by different, ideally it should be a rodent like a mouse, so that eventually that mouse will get eaten by a cat and continue the life cycle. But any creature that encounters those oocysts can be affected, and generally those oocysts in the intermediate or accidental host will spread out through the body. And in doing that, they'll insist, often with no clinical signs, in many of the organs, muscle, heart, but particularly brain. And it's when it infects the brains of different species that it can cause problems. And that's probably the main thing for humans, two main syndromes in humans. Toxoplasma, a lot of people don't realize it's the parasite that is the reason that pregnant women are told not to clean litter boxes, because the fetus, the developing human fetus, is particularly sensitive and can be adversely affected by in utero infection.
And what does toxoplasmosis actually do in the human body? How does it make you sick?
Toxoplasma, ideally in the normal host cycle, it will insist in the muscles and brains, pretty much every organ, but particularly muscle, brain, those organs that are going to be ingested. Or some research now suggests that, or not suggests, but has even shown that toxoplasma infecting the brain will alter the behavior of some of the intermediate hosts. So ideally, if you're talking about a rodent, infection in the brain in which they cause cyst will alter the rodent's aversion to, say, cat urine. And so they will no longer avoid the cat urine, but actually either go towards it and then get themselves in proximity to the host so that they're eaten and continue the life cycle. When you're talking about an aberrant host, you get the same type of cyst. And as long as they stay just a nice, discrete cyst in an organ, they're not really considered a problem. It's when those cysts within the organs bust out, and the individuals, which contain hundreds or thousands of different little organisms, they'll spread out and form new little cysts in different areas or adjacent areas. And it's when those individual organisms are traveling to a new location that they cause a lot of tissue damage and inflammation. And it's that causes either the inflammation in the brain or inflammation in the heart and different organs. And you guys, you could even have discussions, it's kind of like toxoplasma affecting the behavior of animals, and, it's been related to possibly human mental disease such as schizophrenia.
Let me ask you, how do you keep track of all this data that you're gathering from zoo animals that get sick?
As part of the larger invertebrate discussion, the Smithsonian has a huge invertebrate department, and that, you can go on the web and look at all their holdings. They even have their catalogs, I believe, available on the web. But as part of their holdings, in 2014, they developed a cooperative agreement with the USDA to assume responsibility for the National Parasite Collection. And this is a parasite collection that's been developed well over 100 years and includes hundreds of thousands of specimens from all different kinds of parasites. So it's a huge issue, both in curating and taking care of this national parasite collections. Just as with when the USDA was in control of it, it is deemed to be a research asset, and so as such, people add to the collection nationally and also withdraw or request voucher specimens from the collection in doing their different studies. So it's a viable entity and not just something taking up shelf space.
But not every parasite is a multi-host parasite, right?
Okay, yeah, so I think mites are one of those parasites that can have implications for additional types of animals, but most mites, at least the parasitic mites, we often consider them relatively host-specific or at least order-specific. So we had one instance here in an endangered species, and at that time was a critically endangered species, the Cayman Island blue iguana, and it is one of those species that has its own mite species that lives only on that type of animal. In a zoo situation, we often don't like to have animals with parasites, so we try to get rid of them. The acarologist, or the person who identified this mite for us, threw out this question as to whether getting rid of that by default endangered mite, since its host is endangered, was really the right thing to do because you're decreasing that species. And so would you be kind of responsible for the extinction of a species even if it's a parasite? That was just kind of one of those interesting things that you have to think about when dealing with some of the conservation and critically endangered animals.
That is really, really thought-provoking. Thank you so much, Tim. I want to talk about one more group of ugly invertebrates, the spiders. We find them creepy, but should we? Let's first note that most spiders are harmless to humans because they help to control injurious insects. This does not reduce human fear for these small creatures. There are a few spiders in the world that can give painful or even fatal bites. Several highly aggressive large spiders live in the American tropics, and there is a dangerous funnel web spider in Australia. In North America, there is the small, though infamous, black widow spider that has a neurotoxic venom, and about 4 or 5 bites out of 1,000 are fatal. And there's the brown recluse spider, which has a violin-shaped marking on the dorsum of their cephalothorax. Their venom is hemolytic, so produces death of tissue and skin surrounding the bite. Otherwise, the bite is mild, although there are apparently a few unconfirmed deaths of small children and older individuals from brown recluse spider bites. So I guess these few highly venomous spiders out of the 80,000 timid species can be considered pretty bad for humans. Most people are more afraid of spiders than flies, but maybe they have that backwards. Do you know what happens when a fly lands on your food? Houseflies have sponging mouthparts and cannot chew their food. So they puke, spit up, vomit, enzymes onto their food, your picnic plate, to dissolve the food, and then they slurp it up. What a way to transmit disease from one place to another. So we have much more to fear from flies than from spiders. All of that being said, whether we consider a creature good, bad, or ugly, all of them are part of a biodiverse food web that sustains healthy animal lives across our tiny green planet. Maybe all these creatures aren't so bad and ugly after all. Have you ever tried to swim with the fishes? I don't mean that as a threat. I'm asking if you've ever been swimming in the ocean or in a lake and tried to stay underwater with them, watching their amazing command of the water. What makes fish so agile? What enables them to pull oxygen from water for respiration? In this lecture, we'll explore adaptations, biology, and conservation needs of some of the most interesting animals on our planet, the fishes. In the Earth's animal kingdom, there are more than 28,000 species of fishes, a broad designation that includes almost 1,000 species of sharks, skates, and rays, as well as 27,000 bony fishes. I'll talk about fishes to mean 3 of the 7 vertebrate classes, so understand that we group some very diverse aquatic creatures together. The fishes make up almost half of all vertebrate species on Earth, and they're extremely important to the planet ecologically and to we humans economically. Some so-called fish, like jellyfish, are not fish. They're invertebrates. And starfish aren't fish either. I have heard that some people call these invertebrates water awesomes, or the more accepted name of sea stars. These creatures live in the seas, but as invertebrates are not classified as fishes. So let's go back to the true fishes. Zoologists recognize a fish as an aquatic vertebrate with gills, bilateral appendages that occur in the form of fins when present, and usually a skin with scales of dermal origin. The modern zoological concept of fish is used more for convenience than for taxonomy, because those 28,000 living species of modern bony fishes, sharks, skates, and rays I just mentioned make up more species than all other species of vertebrates combined. The ray-finned bony fishes make up over 99% of the total species covered here. Bony fishes evolved about 430 to 440 million years ago in the Silurian geologic period. These aquatic creatures dominate and have evolved adaptations to the world's oceans, lakes, rivers, and streams, from the hottest to the coldest, from the saltiest to the freshest. In adapting to the physical limits of these habitats, fishes have shaped the basic body plans and physiologies that have evolved into the modern fishes we see today. As we discuss the fishes, you will need some facts about the water where they live. Water is really heavy. Pick up a gallon of water or milk. Then imagine the weight of millions of gallons of water in lakes, rivers, waterfalls, or oceans. Water is much denser than air, about 800 times denser, in fact. As one swims deeper, the pressure against the body is much greater and can be more than 100 times greater in the watery depths than at the surface for fishes that swim 2,000 feet or more below the surface. The advantage of this environment is the near weightlessness characteristic of the environment for fish and even scuba diving humans swimming through it. We call this characteristic of an upward force pushing against an object in the water buoyancy. Fish achieve buoyancy through several adaptations. Their bodies are streamlined for movement through their dense watery environment. They can choose to move up or down in the water column just by adding or subtracting air from their swim bladder. The shapes of their fins and tails allow them to move quickly forward or at an angle by using body and tail motion for acceleration and fins as rudders or brakes. Almost all fish have gills for gas exchange. Gills are respiratory organs that we see in many water-dwelling animals and that contribute to gas exchange in a water environment. We see them in fishes, some water-dwelling invertebrates, in amphibians, and even other animals as they develop. Terrestrial creatures that have gills, like hermit crabs, have needed to develop adaptations to keep their gills moist so that they can successfully respire while on land. Fishes have not needed that trait since they live in water. Fishes like mud skippers, which spend part of their time out of water, have evolved moisture-retaining chambers around their gills that are remarkably similar to the chambers we see in land crabs. Fishes have gills that are the most efficient respiratory organs in the animal kingdom, with the ability to absorb oxygen from a viscous medium, water, that has less than 1/20th the amount of oxygen as air. The microscopic structure of the gill and blood capillaries that are close to the membranous surface provide a large surface area for gas exchange with the watery outside environment. The capillaries in a gill absorb oxygen from the watery environment and excrete carbon dioxide back into that environment. The large surface area of the gill available for gas exchange is crucial for the success of this organ, since water contains only a small amount of the dissolved oxygen that is contained in the air we humans breathe. Oxygen has a rate of diffusion into bodily tissues that is thousands of times higher in air than in water. Let's move on from gas exchange adaptations to fish sensory systems. Fishes have excellent senses, which includes a lateral line system that helps to feel vibrations and other movement in the surrounding water. The lateral line sensory system of fishes is made up of modified epithelial cells known as hair cells. These hair cells are stimulated by motion and then send signals to the central nervous system after being stimulated by motion. The fish feels vibrations from prey and other movement due to displacement of water near the body systems, picked up by the fascinating lateral line system. From the physical input of movement to the hair cell's sensory receptors, there is transduction of these signals into electrical signals as the message travels along the body's excitatory synapses, where they finally reach the central nervous system so the fish can respond to the external stimulus. Sharks and their relatives, the skates and rays, make up about 940 species of fishes that have skeletons made of cartilage. Before we talk about their features, let me say this. There are a few species of sharks that are dangerous to humans, as we know. The inshore bull sharks that live near our beaches and rivers, and the pelagic great white sharks that swim the open seas are among them. By the way, you have a greater chance of being killed by lightning or a bee sting than by a shark. Sharks are amazing creatures. Meanwhile, humans are decimating sharks through shark tournaments and for the cruel practice of finning for shark fin soup. That is, a shark is caught, its fins are cut off, and the body is tossed back into the sea. All this for an Asian delicacy that is sold for up to $100 per bowl of this soup. Millions of sharks are being killed this way annually. We fear sharks and we kill them, but the truth is most sharks are timid around humans. Think about that as we discuss these wondrously adapted creatures that evolved before the dinosaurs and have remained almost unchanged since then. The amazing body shape of the shark is well known. Although it looks fearsome with the pointy snout and small eyes, this streamlined form is an adaptation for moving through water quickly. Many sharks have a more streamlined spindle shape or torpedo shape. Sharks have paired pectoral and pelvic fins and dorsal fins, which help with turning ability and stability in the water. They have an asymmetrical or heterocircle tail, which sweeps back and forth to provide forward thrust and uplift when needed. Sharks have paired nostrils on their snouts, or rostrums, which contribute to olfactory sensory abilities of these creatures. Dissolved chemicals in the water are picked up by neuroreceptory cells in the inside of the nares, and olfactory signals are sent from there to the brain. The tough skin is covered with tiny dermal scales, which reduces the turbulence of water flowing along the body while the creature is swimming. These scales are shaped like backwards pointing teeth, so are also called dermal denticles. They're tooth-like and are different from other kinds of scales which provide protection as body coverings. Fish scales are derived from the dermis, and the upper surface of the scales is formed by keratin. Although they come in different shapes, scales are most often arranged on the body to provide overlap from the head to the tail. Sharks are not as fecund as bony fishes like cod, which produce millions of eggs at a time, and their young take a long time to reach sexual maturity. So, this contributes to their inability to rebound from overharvest for human food. Rays make up half of all species in this group of cartilaginous creatures. They're specialized for swimming along the bottom of the ocean or, for freshwater rays, rivers. All rays have bodies that are dorsoventrally flattened, and their pectoral fins have been greatly enlarged to act as underwater wings that, when undulating, propel the body forward. Amazonian river rays are the only rays known to permanently dwell in fresh water, and in this trait, they differ from all other living rays and their shark relatives. These freshwater rays have lost any ability to migrate between freshwater and their ancestral marine environments due to a highly refined osmoregulatory system that allows them to survive in freshwater. Other sharks and rays have evolved physiological adaptations that allow them to osmoregulate in saltwater. Ocean waters are usually saltier than the blood of most fishes. Instead of actively pumping minerals and salts out of their bodies as ocean-dwelling bony fishes do, the ocean-dwelling rays and sharks have evolved to regulate their internal salt concentrations to be the same as the salt concentrations in the water environment outside the body. How do they do this? They maintain adequate concentrations of the enzymes and organic solutes within their bodies in the presence of salty body fluids. Amazonian rays, like the ones at Smithsonian's National Zoo, live in freshwater rivers, so they have problems opposite those of their marine relatives. Remember when you studied basic chemistry and molarity? You would have learned that an aquatic system with some higher salt concentration would tend to gain water if it was adjacent to fluids with lower concentration of the salt. Then each system would be in balance with the other through the transfer of water. This applies to the body of freshwater rays as well. Instead of losing water to an external saltwater environment, freshwater rays have to worry about gaining water because their internal body salt concentrations are higher than the salt concentration of freshwater. The water in which they live has very low salt concentrations, and we know from our understanding of basic chemistry that water will seek to equalize salt concentrations across cell barriers. One result of the diminished need for salt excretion is that the freshwater river stingrays of the Amazon basin no longer have a need for the rectal salt excreting glands found in saltwater rays that eliminate excess body salts. So in Amazon rays, these structures are vestigial. That is, the organs are reduced in size and are also no longer capable of secreting salts from the body system. Amazonian rays have been isolated from salt water for so long that they have also lost the ability to retain urea salt. This adaptation requires the Amazonian rays to sever ties with their ancestral ocean environment, so they are freshwater rays in our modern era. Freshwater and ocean rays all have 2 rows of 5 gill slits on the bottom or ventral side of their bodies. They also have 2 modified gill slits on the upper or dorsal side of their bodies, and these are called spiracles. Their spiracles are located behind the eyes on the top of the body. Since the ray's mouth is often on the watercourse bottom at or below mud level, water enters through the spiracles on the dorsal surface, and this prevents the gills from being clogged with silt, something that is important for efficient respiration. This anatomical adaptation allows rays to breathe more easily while they're hiding in the sand and mud. The rays have long, whip-like tails that are armed with 1 or more spines, and these spines have venom glands at their base. The spines can seriously wound or even kill a predator or a human, and the wounds can take a long time to heal. Most fish, the sharks, rays, and bony fishes, are ectothermic, which means that their body temperature is the same as their environment. For most of these species, any heat generated internally is lost to the heat-absorbing water. However, zoologists have found that some fishes, like great white sharks, maintain temperatures higher than the local water temperature in their internal organs and muscles, perhaps even 10 degrees Celsius warmer. Marlins and other large pelagic relatives, that is, open water dwellers, maintain high temperatures in their brains and eyes in what researchers have called regional endothermy. In this kind of thermal regulation, heat is generated in marlins by a special heat-generating organ near the brain. This heat is conserved by a parallel bundle of blood vessels and counterflow system called a rete mirabile. The heat organ itself has been examined, and its heat-producing cells have special physiological characteristics that control calcium metabolites in the body region and are apparently derived from muscle cells. High temperatures lead to faster swimming because warmer animals move more quickly, and the regional high temperatures in the body may speed the digestive and nervous system in these fishes, among the fastest in the seas. The coolest new research about fish might be a discovery published in 2015 about the rather tubby, 3-foot-long opa, which lives in oceans around the world. This colorful fish lives 150 to 600 feet below the surface, where the water temperature is about 50 degrees Fahrenheit, and it uses its massive pectoral muscles to help it generate warmth for its brain, eyes, heart, and body. Biologists with the National Marine Fisheries Service caught opa on hook and line, attached short-term monitoring devices to them, and were able to take the temperature of the world's first known warm-blooded fish. They learned that, unlike most fish, which are the temperature of the water they swim in, the opa can be 20 degrees Fahrenheit warmer than the surrounding water. Not only do these fish produce extra heat with their pectoral muscles, they help to conserve this heat through a really efficient rete mirabile system of capillaries in their gills. The rete mirabile countercurrent heat exchange system enables warm blood moving toward the interface with the cold ocean water to leave its heat behind, and cold blood returning from the gills to be warmed as it moves close to other warm blood, leaving much of its cold behind. The OPA also has adaptive layers of fat that act to insulate its heart and pectoral muscles from the cooling effects of cold ocean water. This matters because warmer fish can expand their range in both latitude and depth, swimming better because of their warmth. So, although little is known about this pretty fish that doesn't look like much of a predator, scientists think its adaptations allow it to be a good predator in the ocean because of its acute vision, rapid response times, and endurance. And that's pretty cool. Economists have warmed to fishes and the importance of conservation fisheries. For fishing in the United States, the U.S. Fish and Wildlife Service put together a report called Net Worth that estimated fishing in the United States to be worth over $3.6 billion per year, or $10 million per day. That is a lot of trout, salmon, bass, pike, and other bony fishes. Climate change may affect these fisheries in a negative way. Despite the good historic and current efforts of licensed fishermen and other conservationists who help